Good afternoon, ladies and gentlemen, distinguished guests. Welcome to join us at the Laureate Lecture of 2022 Tang Prize in Biopharmaceutical Science. I'm Angela Liu, your MC for today's event. To begin with, we would like to invite to the stage the host of this lecture, Professor Wang Huijun, academician of Academia Sinica, to give us the introduction. Ladies and gentlemen, please welcome Professor Wang.
Good afternoon. I notice this event is executed in military precision. So there was a countdown of 10, right? So today I'm allocated for 10 minutes. So I'll be sure I will finish this introduction in 10 minutes. Now today, it gives me great honors to introduce today's laureates of 2022 Tang Prize in Pharmaceutical Science. Now, today, I think you all know that we have three laureates, Dr. Katalin Kariko, and Dr. Drew Weissman, and Dr. Pieter Cullis. And all three of them are, of course, distinguished scientists globally. And today, it's an event that was really remarkable in the sense that different kinds of expertise have been joined together to develop this remarkable vaccines that save millions, truly millions of lives. And the start of COVID-19 in the late 2019 and early 2020s sort of struck us with great surprise as well as shock. And we all know that to counter this infectious disease, a bit unknown, and that requires all kinds of tools to counteract this infectious disease attack. And so now, of course, drugs, antibodies, and so on are valuable tools. Nonetheless, I think we all, for the global consideration, we all need vaccines, as we all know. So early on, the vaccine development was kind of a bit slow, using a more traditional technology. However, I think in very short time, some of the idea of this early concepts of messenger RNAs might be used to become a vaccine has emerged. And fortunately, I think based on the remarkable contribution of those three scientists that we're going to hear their talk shortly, they gave us an opportunity to rapidly develop this vaccine. Of course, many governments around the world have helped in contributing these advances. Now, like in the United States, I understand there was a project WARP to pump in a huge amount of money to facilitate the success of this. And so I think with this, combining all the basic science and translate it into the truly useful technology and products to cure, or not cure, but to prevent the infection of this kind of all this, a truly unknown enemy, that give us really a powerful tool. Now this mRNA technology, as we will hear shortly by the three outstanding speakers, gave us this opportunity to develop the vaccines, applicable vaccines in just within 1 year. And that is a truly remarkable and significant contribution to the world health. Now, today, the three laureates. I think you have been hearing their introductions and so on, so I won't go too much into the details, but suffice to say, I'd like to say that they will give us 3 consecutive lectures, and there will be no interruption between them. And so the first speaker, Dr. Kariko, will give us a description on developing mRNA for therapy. The second talk will be given by Dr. Drew Weissman, and he will discuss the nucleoside-modified mRNA-LNP therapeutics. Okay. Then finally, I think Dr. Pieter Cullis will give us some concepts and the progress of the development of this LNP system. So his talk will be on design of lipid nanoparticle systems that enable gene therapies. And so let's welcome those 3 speakers, and we look forward to their wonderful talks. Thank you.
Thank you very much to Professor Wang for the introduction. Please take your seat. And first of all, we would like to invite Professor Katalin Kariko to the stage to deliver her speech. Welcome.
Thank you very much. It is wonderful to be here and accept the award together with my colleagues, Drew Weissman and Pieter Cullis. Today I talk about how the mRNA was developed for therapy. I have to say that RNA was discovered in 1961, and it took 60 years when finally mRNA became an FDA-approved product in the form of COVID-19 mRNA vaccine. And during these 60 years, a lot of things happened. So those who think that the vaccine was developed overnight is mistaken, because I would say every 20 years something happened. So the first 20 years was structural, functional characterization of the mRNA isolates. People couldn't make RNA as we could do today. In 1984, we could make mRNA, and it was another 20 years when scientists made different type of mRNA coding different proteins, and then those were tested out in cell culture and in animals. And it's about like 2000 when the first RNA company was formed, and that was, again, in the 20 years when different human trials started. So this is what I'm talking about. So 1961, when mRNA was discovered, it even started the clock. In 1961, when mRNA was discovered, and 2 papers published in Nature and describing this unstable intermediate, this middleman, which carries the information from the DNA to the protein synthesis place with the ribosome, and the protein is made, and the protein can be an enzyme, produce lipids and other things, and that's what we are. So the mRNA was a very short-lived product. So at the beginning, the scientists couldn't synthesize the mRNA, they just could isolate. And then they looked at there. So scientists discovered that the RNA has a long poly A tail, and then immediately invented purification. So in 1972, oligo-DT cellulose was introduced, and RNA could be purified. So using this purification procedure, other scientists look at the 5' end, the cap part of the RNA, and 1975, many papers in Nature published about what is this structure all about. And they already showed that if the cap is not present in the mRNA 5' and the mRNA is not translating. So a lot of discoveries was made in the 70s. Other scientists look at the functional characterization of the mRNA. Even in the year when it was discovered, 1961, they already showed that they can translate. But here scientists started to use reticulocytes, the precursor of red blood cells, and then, because those are enriched with globin mRNA. So they tested out in rabbit reticulocyte lysate, they injected in frog oocyte, or they used liposome. And they enrapted the liposome, and then they fused to primary mammalian cells. And they demonstrated that the mRNA, when it was different delivery were used, they code that critical protein. But it was all the time they just could use, the beta globin coding protein. So what happened in this kind of time, I was an undergraduate student, and I get the opportunity to work in the lipid laboratory. And the supervisor, Erne and Eva, tried to make a liposome because they were reading this paper, which was Dimitriadis and Ostro published in Nature in 1978. And because I was in the lipid lab, I helped them to isolate phospholipids that used for liposome. And we did not deliver at that point RNA, we delivered plasmid DNA. And we were very successful on that. And it is very important for all of the students to start something somewhere and then later, think about a lot what was done. But that time I was undergraduate student, and one day, Thomas walked in the lab, and that's how I went to the RNA lab, went from the lipid lab to the RNA lab. So I was not that smart to figure out that one day this RNA and the lipid belongs to each other, but it by just happening by chance. So he actually synthesized some of those cap structure for those scientists who discovered in 1975. But here my responsibility was to synthesize this short oligonucleotide, which had an antiviral effect. I worked together with János Ludwig, and even today, he's an organic chemist, even today, I ask his advice. So please be kind to your fellow students, because you don't know how much you need their help one day. And this is also important, that when we collaborated, I was the biologist, set up the antiviral lab, because this small molecule was responsible for antiviral activity. It was naturally, this molecule is made by oligoadenylate synthase, and that when this molecule is made, activates RNase L, which then can, that was the idea, cut up selectively the viral RNA. As it turned out, it's not selective. Anyway, this was the first time. It was 1985 when I lost my job. And I have to say I wouldn't be here today if at least 4 times I would not lose my job. So things can be, it depends on your decision that what do you do after your position is terminated. So I went to the United States, and Professor Suhadolnik himself was also working on these 2', 5' molecules. And actually, he wrote the book on basic, nucleoside-modified nucleosides. So I worked in the lab, and so we did more of these 2,5-A compounds, but we could not formulate well that it would have a good antiviral effect. So what happened is, in 1986, when HIV was the major viral problem, eventually we couldn't use this antiviral compound, this short 2', 5' compound, but we used the double-stranded RNA, which was mismatched, it was less toxic, and we treated the HIV patient with this double-stranded RNA. So as you can hear, wherever I was working with RNA, different enzymes that modify the RNA. And this segment, now that I was in 1985, is when already scientists discovered how to make RNA by design. And this happened in the Harvard laboratory. Tom Maniatis, Douglas Melton, Paul Krieg, Michael Green, they discovered that if they use phage RNA polymerase, and from plasmid DNA with the promoter and adding all of the basic 4 nucleotides, capping enzyme, they can make an RNA which has all of the structural element and coding for beta interferon. When they injected this mRNA to the frog oocyte, what they found is that the culture medium where these injected eggs were incubated had antiviral effect. So this was the first time that scientists could show that messenger RNA made in the tube has translating functional protein. So a lot of people get inspired by this discovery, and you could see that in the 1990s, every year there was a paper out on mRNA. In these days, every hour, more than 10 papers is out. But this time it was easy to follow the literature. I was mostly inspired this 1992 paper where it was shown that mRNA coding for a therapeutic protein, vasopressin, and injected to the animal, which had missed and didn't have enough of that, they could treat the animal with that. So for that on, I set up that I will do mRNA coding for therapeutic protein. But you can see that 93, 94 already people started to use mRNA for vaccine. And 95, 96 started to use for cancer vaccine. So infectious disease vaccine, 93, 94 for influenza, 95, 96, it was for cancer vaccine. So, but those scientists didn't follow up. And the reason was because the protein was made from these RNA was very small amount and was made in very short period of time. And it degraded, and later we show also that it was immunogenic. So a lot of people, those even who I was inspired by, never published another paper on that field. So what you can see is the following years is that the mRNA was continuously improved, and the formulation, the lipid formulation was improved. And many of these steps were when the RNA was with the lipid formulation was together advanced and made the mRNA more translatable. And eventually we come to the end when it was lipid nanoparticle was used to deliver mRNA. And so this was needed, the delivery and the mRNA improvement. mRNA was purified and so on. So I started to work on mRNA in 1989 when I joined the University of Pennsylvania, where here my colleague Alice Kuo, she was actually graduated from the high school I visited today, this morning, and also the university, the National Taiwan University. And 2 of us, or 3 of us, started to work and develop mRNA for therapy. Why I am showing this urokinase receptor example here, because this was Elliot Barnett, who is a cardiologist, was interested in this molecule to make sure that in the blood vessel, the blood will not clot. And this molecule was only, this protein only was functional if all of these post-translational modification, glycosylation, GPI, anchoring, other things were happened, and it was modified. And voila, we delivered the mRNA I made in vitro, and when we delivered to the cell, we could see that the receptor was functional. So the cell knew what to do, how to further process. So this gave us inspiration. But it happened again that, I kind of lost the position. I was demoted and ended up in neurosurgery, where I spent 17 years. Again, David Langer helped me, who was in the prior lab, was an obnoxious student, but I was kind enough to him that when he learned I lost the job, he helped me to get this one. So be nice to everybody there. And then here is this laboratory in the left corner. This is where I worked 17 years. And yes, so I did not have really lab coat and things like that, but for RNA, you have to be very careful. So what we did, we tried to develop mRNA for therapy, mostly for stroke treatment. And when we did all of these studies, like we tried to deliver nitric oxide synthase enzyme to dilate some blood vessels and others, when we injected to animals in the brain or others, we did not find that any problem would be with the RNA, whether it would be immunogenic, especially because the RNA coded for a protein which was already present in the body, in these animals, and I was not thinking that there is a problem up until I met Drew Weissman. Really, we met at the Xerox machine as it was described, and Drew was interested to make vaccine. I was interested to make therapeutic protein, and so, but, you know, we started to work together, made the gag mRNA, and he tested out on human dendritic cells, and he concluded that it is a great vaccine because it codes for the protein, it also activates the cell, and he measured high level of inflammatory molecules were secreted by these cells, which I was not very happy about because I want to develop for therapy. So we asked the question with Drew that why this RNA is immunogenic, why it increase tumor necrosis factor alpha, for example. And if this RNA is the same, you know, which we have in all of our cells, but then the idea came that maybe the reason is because we are adding to these immune cells, these human dendritic cells, from outside, and maybe that's the reason. And maybe all of our RNA inside our body is immunogenic if it is coming from outside because the cell damaged or something. So we set it out that we will investigate why inflammatory, and we isolated RNA from mammalian cells and different compartments, and what we tested out similar way, and what we found is that this RNA is, you know, the purple shows what I made in vitro in the tube, and those blue is the different compartments where E. coli RNA shown in red. But what was interesting is that transfer RNA, when we delivered, were not immunogenic at all. And because the transfer RNA has a lot of modification, of course, many ways is different from the in vitro transcribe because it is shorter, it is different 5' and 3', but I'm just, for the shortness of the story, I'm saying that a lot of modification are there, and 25% of the nucleosides are modified, so that the idea came that could it be that these modifications, one of them, all of them, may be contributing that the tRNA is not immunogenic. So investigate that. I just showed this one to scare you a little bit, but how many modifications are there? And we were wondering that how we could test it out, because even the enzyme introducing these were not known. But the only known was that how 2' methylation and pseudouridylation actually happening in the RNA, and actually discovered by a fellow student from University of Szeged. We lived in the same dormitory, and again, it is important not to know just only that Tamás was a good soccer player, but what he did after he finished, graduated. So he was working with RNA and discovering these things, and so I asked his help, you know, to modify the RNA, but at that point, in vitro, with this method, we couldn't do it. In nature, all of our RNA is made from the 4 basic nucleotides, and post-transcriptionally, modification are introduced by enzymes. So we couldn't follow on this one, decided that maybe we have to purchase, or how nature is not doing, but we have to purchase nucleoside triphosphate, which already the nucleosides are modified, and hoping that this RNA polymerases will incorporate these to the RNA. So we were insisting that we are just using modifications that are already present in the human body, so not anything which is naturally not present. And these 10 nucleoside triphosphate modified, one was commercially available. And we purchased that, and we tested out. 5 of them could not incorporate, but 5 of them could. And so we generated this mRNA shown in this agarose gel. Then we tested out in the same way. We did prior to these experiments when we isolated RNA used. And what we found is that in blue are now showing that those which was not containing any modified nucleoside. But then in orange, you can see different modification still induce high level of TNF-alpha. Those were still inflammatory, but more importantly, some of them were not. So when we looked at closer, we found that all of them, which was not inflammatory, contains modified uridine. So we concluded that seems like nature selected uridine to be recognized as a extracellular RNAs coming towards an immune cells, and if we modify it, then we don't get activation. But of course, this was just one goal, that not to activate. We also want to use it to code for some therapeutic protein or antigen, so it was important to see that whether we can translate if the RNA, messenger RNA made with this modified one. Like one of them, the 2-thiouridine, were not translated at all, but most importantly, the pseudouridine containing RNA, which is in orange, is translated very well. So well that 10 times better than the conventional uridine containing RNA. So it seems like, a dream comes true, because now we have an RNA, which messenger RNA, which is not inflammatory, and 10 times more protein can be made from it. So what happened here in the United States, usually that you file a patent, you publish, you establish, as we did with Drew, establish RNA-Rx, made a business plan, and we submitted that we will use erythropoietin-coding RNA, and we demonstrate that it can increase the hematocrit and the reticulocyte level and the hematocrit in animals. And so that's what we did. In 2012, we published that when transit complex pseudouridine-containing RNA was injected to the animals, in the mice, intravenously or intraperitoneal sometimes. Here, what we found is that when the erythropoietin-coding RNA was containing pseudouridine, this is in red in the left panel. You can see that 4 days we could measure the protein circulated in the blood. And when we had the conventional unmodified RNA, and these are very small amount, 0.1 microgram. If you work with RNA, you would know that, or nucleic acid, how small amount. But when it was not modified, you can see in the blue that it disappeared quite quickly. The half-life is, the erythropoietin is 2 hours. So it meant that when you injected pseudouridine-containing RNA, then it had to be continuously translated, so the mRNA was still there at day 4. And the right panel shows that the produced protein from this RNA was functional because the hematocrit increased, so the animal had higher red blood cells. And when weekly injected, then we could maintain high hematocrit. And in the middle panel shows that when we use pseudouridine-containing RNA, there were no interferon was induced in those animals. So with that, it seems everything is going well, but that was 10 years ago, when last time I lost my job, and then I had to move on. So here I am talking about now the last 20 years for this scheme, because CureVac was established in 2000, and 2008 was BioNTech, and 2010 it was Moderna. So the animal studies finished, or some is still follow-up, and then clinical started. So as I mentioned, 2013, when I lost my job last time, I went to Germany and joined BioNTech, established by Ugur Sahin and Özlem Türeci. And at that point, we were not in this fancy building shown in the right corner. We were on the campus, and we were just a handful of people. But what happened there, we further optimized the RNA. We did codon optimization, optimized the UTRs and the cap. We introduced the cap1. We did purification, and all of these modifications. Eventually, what it resulted, that now is not a short period of time, very small protein was made. This was significant amount of protein was made quite long time. And so with this one, the optimized mRNA was ready for prime time. Why it is important to talk about that this mRNA is so important? I have to say because the protein, therapeutic protein, which was FDA approved, it is the this is the largest segment of the approved product, and this is more proteins are approved, and they are fastest growing group of therapeutics. And there are many different kind of proteins. Actually, the first protein was insulin, which was introduced 100 years ago, 1920. It was purified from animal pancreas. And then it was 1982 when a recombinant protein, the first one introduced, actually, it was also insulin. And then those proteins, which is recombinant, is quite expensive because it had to be purified, and during purification, sometimes changing the protein. So it is, what it is very expensive. And the idea is that if we deliver the mRNA, then the human body can generate the protein. And so it would make it cheap, and it would be more reliable. So when I was at BioNTech, so we started to use, because now that the RNA translated very efficiently, so we generated bispecific antibodies. The bispecific antibodies is, one arm is seeing the tumor-specific protein on the surface of the tumor cells, and the other arm is seeing the immune cells. And we generated these kind of antibodies and tested out in animals how we could eliminate actually already established large tumor using this approach. Highlighted in yellow, you can see that these are already these bispecific antibodies, mRNA, they already entered to the clinic. Actually, conventional RNA, conventional antibody coding mRNA also entered. So this was followed up by another studies where mRNA now is coding for 4 different cytokines listed on the lower left corner. And these mRNAs, when we injected to animals that had like melanoma, in this case of mice showing here, and what happened is that you could generate an abscopal effect. So the injected tumor is, which is at that point is cold, but these cytokines will help the different immune cells to run to this tumor and figure out how to recognize tumor cells. And then when they circulate around the body, they can eliminate, as you can see on the right panel, that eliminate tumor from the lung. And we started these studies together with Sanofi, and you can see here that also is in clinical trial. So this idea is that there are cancer vaccines, but these are not vaccines because the mRNA is not coding for cancer antigen, rather it codes for cytokines. And in the case of antibodies, what I mentioned again is not cancer vaccine, it is the mRNA codes for antibody recognizing cancer-specific proteins. So what happened now? We are in 2023, and now that we can see that more than 150 clinical trial is ongoing. These are to prevent or treat different kind of disease with mRNA. And probably Drew Weissman, colleague, will talk about how many different vaccine is during development for infectious diseases. For influenza, actually, BioNTech and Pfizer started together working on 2018 to develop mRNA-based influenza vaccine, which in 2020 put in the back burner because the COVID had to be developed. But this both for Moderna and BioNTech has a phase 3 trial. RSV is also close to approved by that Moderna is developing. HIV, there are both Moderna and BioNTech has a program for that. Zika virus program also, so it is clinical trial. Herpes simplex, this was by Penn and together with BioNTech. EBV, CMV, Nipah, both, all of these were antiviral vaccine is developed. Then also bacterial vaccines, so for tuberculosis or Borrelia, which causes Lyme disease. And this is a program, Borrelia, developed by Moderna. And malaria, which is a parasite, that kind of vaccine is also in development. The other thing is the cancer vaccine. All of these companies, Moderna, BioNTech, and CureVac, who were the first three ones there, they all established a cancer vaccine program. And that was the main goal, and learn many things. And there are programs where the mRNA codes for shared tumor antigens. And these are from the patient sample, identify what kind of tumor antigen present in the patient samples. And those are fixed treatment. And there are also vaccines where the patient tumor are removed, the sequence identifying neoepitopes, those are which resulted from mutation, and then messenger RNA is generated, which codes for these neoepitopes, and these individually for just that patient can be used, and they inject. And at present, the success for cancer vaccine, I can say, is for Moderna is for the melanoma trial, and for BioNTech is the pancreatic cancer. It is very critical that in both cases, the tumor was not spread, and the tumor was removed, dissected, sequenced, and then the vaccine were generated. And the epitope for Moderna had 43 different neoepitopes they used. And then they used the patient and they vaccinated them. And what they could find is that the tumor return was delayed very significantly. So this is how successful right now. But more research is needed for that. Acute diseases, VEGF-A mRNA, relaxin mRNA for heart failure. Actually, VEGF-A mRNA was used 2018 already with AstraZeneca and Moderna. So these were well before 2020 when the COVID happened. And VEGF-A mRNA is this VEGF-A, which was coded by this mRNA, generate new blood vessels. And these studies first started to treat diabetic patients' necrotic wounds. And they could see healing accelerated. And later, these mRNA were injected to patients who were undergoing bypass surgery. And they injected naked RNA to the body of the heart. And they could see that heart perform much better. There are many genetic diseases which started to use mRNA for treatment. Methylmalonic acidemia by Moderna reported success with treatment, where the mRNA coded for this protein, which is missing or very low level present in the patient. And more success we could see when mRNA is coding for the Cas9 enzyme, which recognizing with the guide RNA specific part of the human genome where problems happen. And this was, one of them is the amyloidosis, and other is angioedema, and which maybe soon will be FDA approved, will be the sickle cell anemia, when the mRNA targeted by this guide RNA to specific part of the chromosome where they interrupt. They are not incorporating new genes, or it is just only interrupting a gene, which, whether it is making a faulty protein, or whether, in the case of sickle cell anemia, the BCL11A, that if you interrupt, then you won't generate beta globin, but it will be gamma globin. So this is where it is. And then more studies are coming for treating allergies. These are peanut allergy, dust mite allergy, and it can induce tolerization. At BioNTech, we did studies where we also use for autoimmune disease inducing tolerization, and all of these studies need more animal studies to enter to human trial. And what I am doing now, I try to inspire the new generation. So I mentioned today I was in the high school, and here is with Drew and me doing some experiment, how we are presenting ourselves to the children. And I also wrote a book about my life experience. And with that, I want to also thank to all of my colleagues who I work together, especially at BioNTech. And I am also happy to thank to my family. My daughter who is a 2-time Olympic champion, and my husband who supported me always in my studies. And thank you very much for your attention.
Thank you very much to Professor Kariko for your speech. Please take your seat. Thank you. And next, we would like to bring up to the stage Professor Weissman to deliver his speech. Welcome, Professor Weissman.
Thank you very much. So what I wanted to do today is not spend a lot of time on heavy-duty data, but talk to you about a few different aspects of RNA vaccines and therapeutics. First, I wanted to explain why RNA vaccines work so well. Hopefully, everybody in this audience has had numerous RNA vaccines put in their arm. I apologize for the pain and discomfort, but that just tells you it's working well. But what people don't understand is why the vaccine works so well. And then after that, I wanted to talk to you about the future of mRNA therapeutics. And I'll talk a few different aspects of specific delivery. So I'll quickly go through this. Katie mentioned it. RNA was first injected into an animal in 1990. And I bring this up because I speak with a lot of lay audiences. And what I try to explain to them is what the RNA vaccine is and why it works so well and why they need to take it. And I'm guaranteed to get one question from every audience, which is, well, I'm nervous because it was invented in 10 months. And I don't trust something that was invented so quickly. It wasn't invented in 10 months. mRNA was first found in '61. It's been in animals and people starting in 1990. But after that first injection, it was studied for vaccines. And that was for a number of reasons. Principally, there are 17 different innate immune sensors. So these are germline-encoded proteins in all of our cells that produce a sensor that recognizes RNA and signals that it's foreign. And there's a number of different signals that are induced. It makes inflammatory cytokines. It makes type 1 interferons. It induces apoptosis and cell death, and it directly inhibits RNA translation. So Katie showed a version of this slide. When we were trying to figure out why was RNA immunogenic, we had the idea of, well, let's test a bunch of different kinds of RNA. Because at the time, none of these toll receptors or other sensors had been identified other than PKR and OAS. And we wanted to understand, well, is it simply that RNA is inflammatory no matter how you give it to a cell, or is there something special about the RNA? So we divided up cells, both bacterial and mammalian, to different types of RNA, and we measured their inflammatory activity using dendritic cells. And we chose dendritic cells because they have most of those 17 innate immune sensors. And as Katie mentioned, what we found is that, it doesn't work, the tRNAs had no inflammatory activity, while bacterial RNA was highly inflammatory. And that led to a hypothesis that maybe nucleoside modification and the level and the type that's in the RNA determines how activating it is to our cells. As Katie mentioned, there's over 100 different types of modified nucleosides. It includes things like isomerizations, methylations of both the base and the sugar. Recently, M6A was found to be reversible and involved in controlling translation. So we tested nucleoside-modified mRNAs, as Katie mentioned, and found that certain modifications got rid of most of the inflammatory activity. But that wasn't the striking part and the unexpected part. When we tested those modified RNAs to see were they still translated, and we had a bet at the time, we of course lost that bet. Not only was the modified RNA translated, but it was translated much better than the unmodified RNA. Prior to this, every RNA delivered was either purified from cells or in vitro transcribed with polymerases. So the addition of nucleoside-modified modifications got rid of the inflammatory activity and greatly increased the amount of protein being produced. So this is now early 2000s, and I was still going to HIV meetings, and I was seeing people like Tony Fauci, who I did my fellowship with, and Gary Nabel, who I knew from the NIH, and other luminaries in the science field. And they would ask me what I was up to and what I was doing, and I would sit down and explain what Katie and I were doing with mRNA. And they would smile and they would nod, and then they would say, Drew, you know, why don't you give this up and do something useful with your career? You're wasting your time. RNA has failed clinical trials. It's never going to work. It will never make enough protein to be useful. You should just give it up and do something useful. And I would say, well, you know, let me try and explain why we're interested in this. Therapeutic proteins are one of the most advanced and advancing fields in pharmaceutical science. The problem is to make a therapeutic protein, you start with a 50,000-liter drum of CHO cells or other cells that make your protein. Then you have to figure out how to purify that protein away from cell culture contaminants. So what that results in is that the cost of a monoclonal antibody is typically $50,000 to $100,000 a year. That limits its usefulness. Katie and I would argue if you gave mRNA that encoded the protein, it was much more potent, it was easy to make, there were no CHO cells involved, there were no 50,000-liter drums, and it was probably safer because instead of relying on a tumor cell to fold and modify your protein, you're using host cells. We're the factory for protein production. They listened to that and smiled and walked away. So the first thing that we looked at, and this had been an interest of mine since leaving the NIH, was developing vaccines using mRNA. And we put together a vaccine, and this was early on, 2016 or so. And in this case, we used hemagglutinin from influenza virus. And everybody here hopefully has had influenza virus, and you know that every year you have to get a new vaccine because the virus mutates. So the hope was, well, can you develop something that's broadly protective? And that's a goal of my lab and many other labs. But the first thing we did is we made a very simple vaccine. We put influenza HA in the mRNA. We encapsulated it in LNPs that Pieter will talk about, and we injected mice. And we did a comparison. So this is the mRNA. This is an activated virus, which is what us old folks get as a vaccine every year. This is live virus delivered to the nose, which is what we give our kids every year. And what was striking and what caught everybody's attention is that the RNA-LNPs were 50 times better than an activated virus and 5 times better than live virus. Now, when you talk to a vaccine manufacturer and you show them a new vaccine that doubles your titer, they get excited. That's a significant increase. So when we showed Ugur this, and we showed him a 50-fold increase in titer, he got very excited, and he funded our lab to develop vaccines for BioNTech. But as immunologists, we wanted to understand how this vaccine works. And this isn't described very much. You read in the New York Times and in our other famous publishing sites, the vaccines work well, but nobody really knows why. The way a vaccine works is that it does two things. It stimulates B cells to make antibodies, and it stimulates T cells to give help to make the antibody production better. There's a particular type of CD4 helper T cell that stimulates antibody production. It's known as a T follicular helper cell. And what this cell does is it forms germinal centers. In the germinal center, the TFH interacts with the B cells that are learning how to make antibodies. And they stimulate them to proliferate, to affinity mature, to produce long-lived memory and high levels of plasma cells that secrete antibodies. So what we wanted to know was this vaccine working through a specific induction of TFHs. To do that, we used a macaque model. So the specifics of the data aren't important, but the range of the numbers are. What we're doing here is we're using flow cytometry to measure antigen-specific TFH cell induction. So what we've done in these macaques is we've looked at them before they're immunized. We immunize them with an HIV envelope protein with a double-stranded RNA adjuvant. Double-stranded RNA is thought to be the most potent type of adjuvant for inducing TFH cells. And then we immunize with mRNA-LNPs. And as you can see, the mRNA-LNP vaccine gave enormous TFH responses compared to the best adjuvant out there. We've gone back and done calculations. So for your standard vaccine using alum or MFA or other FDA-approved and unapproved adjuvants, typically 5% of the CD4 helper response is TFHs, and that's considered a good response. With mRNA-LNPs, it's over 50% of the CD4 helpers are TFHs. And as I showed you before, TFHs drive antibody responses. This is the mechanism for why you see antibody responses that are 5 times better than infection in COVID-19 people. So let me now turn to the future of mRNA therapeutics. My lab and other labs, including Moderna and BioNTech and many others, continue to make a variety of different types of vaccines. Katie showed you some of them. We've got 9 different vaccines in phase 1 clinical trials right now, including for things like HIV, hepatitis C, C. difficile, norovirus, malaria, TB, and many other diseases. And this number is going to keep increasing. But what else can you do with RNA? Well, the original approach that Katie and I were thinking about was using RNA to deliver therapeutic proteins. And of course, the inflammation caused that to be derailed for a while. But since the inflammation was solved, we and many others are looking at delivering RNA to produce a therapeutic protein. Now, anybody who does nucleic acid therapeutics tells you the problem is delivery. How do you get the DNA or RNA or virus to the cells you want? Pieter will tell you that LNPs go to the liver. They also go to dendritic cells, which is why you get a good antibody response. But my lab was interested in figuring out how can we target lipid nanoparticles to other cells, tissues, and organs. And we've started off concentrated on 3 main sites. One were endothelial cells, the vessels that line every organ in our body. The next were T cells, and the last were stem cells. But how do you target these LNPs? LNPs are made of 4 lipids. They have a small amount of PEG on their surface for about half an hour after injection, and then they're rapidly taken up by liver or dendritic cells. We asked a simple question. Could we add a targeting ligand to the surface of the lipid nanoparticle and use a lipid that allowed it to stay with the lipid nanoparticle in vivo? And we used a variety of chemistries and a variety of antibodies or pieces of antibodies or ligands for receptors or other types of proteins. We're looking at mini proteins. We've looked at aptamers. So you can use any kind of targeting molecule and attach it to the surface of the LNP. When we did that, using electron microscopy, the morphology of the LNPs didn't change. They got a little bit bigger. That's about the thickness of an antibody coating. Most importantly, they retained function. They could still deliver the RNA. So these are PCAM cells, and these are UVEC cells, and UVEC cells express PCAM. When we delivered LNPs that were not targeted, the UVEC cells didn't take them up and no RNA was translated. When we added an antibody against PCAM and added them to UVEC cells, nearly 100% of the cells now took up the LNPs and translated the RNA. So adding an antibody to the surface of an LNP targeted to a specific cell type, and it retained activity. So we've studied a variety of different targeting systems with a bunch of different therapeutics in mind. One of the first things we were interested in were T-cell targeting. And we did this because I come from Penn. And if you know Penn clinically developed CAR T-cells. The first 2 CAR T-cell drugs were developed and FDA approved by Penn. The problem with T cells and therapeutic nucleic acid delivery is that they have no endocytic activity. They cannot eat particles. Dendritic cells are great at it. Endothelial cells are good. Just about every cell in the body can eat particles except T cells. So our idea was if we were to target T cells in vivo, we would add an antibody that binds an antigen that endocytoses. And that way, we would bring the LNPs into the cells and get translation. And that's what we see here. So ibalizumab is an FDA-approved anti-human, anti-CD4 antibody. And we added it to the surface of LNPs. For 293 T cells, it didn't matter. For HeLa CD4 cells, there was a little bit of an increase. For purified human T cells in culture, without targeting, we got nothing. With targeting, we got very high levels of RNA delivery. So this told us that the targeting could target CD4 positive T cells, and it could result in RNA uptake and translation. We injected LNPs into mice. Here they're targeted to mouse CD4. And we saw an increase in splenic delivery. Spleens have about 20% of their T cells are CD4 positives. But most interestingly, we re-imaged the animals after we removed all of those organs. So what we're doing here is we're delivering luciferase encoding mRNA. Luciferase is a protein that was originally derived from fireflies, and it's the protein that makes fireflies glow at night. So you can use this protein, or RNA-encoded protein, to identify protein delivery. And we're doing the same thing here as a firefly does. We add substrate, ATP, and we stick it under a microscope that measures light production. And what we found in these animals is that with CD4 targeting and all of those organs removed, we still had activity in lymph nodes. These are para-aortic and inguinal lymph nodes. So that means with an antibody-targeted LNP, we could deliver those LNPs intravenously. They would escape the bloodstream, go into the tissues, into the draining lymphatics, to the draining lymph nodes. There, they would target CD4 positive T cells, be taken up, and the RNA was expressed. So we switched to a model system. And this system uses an enzyme known as Cre recombinase. And Cre recombinase looks for specific sites known as LOXP sites. And what you do is you put LOXP sites around a stop codon. So if you deliver Cre recombinase, the stop codon is removed and you turn on the gene. And usually you put it in front of a fluorescent protein. So any cells that take up the RNA and the Cre recombinase is delivered turn green or red or whatever the fluorescent protein is. So we did this. We delivered targeted or untargeted LNPs with the Cre recombinase enzyme. And what we saw is when the cells were targeted, we got high levels of gene recombination, which equals green color, in the CD4 positive T cells, in the spleen and in lymph node. Now, my lab started as an HIV lab and still does a lot of work in HIV. What we wanted to know, though, is what cells are we targeting? And what we've done is we've added an activation marker here. This is CD25. We've looked at other activation markers. And as you can see, the activity is equal in resting T cells as well as activated T cells. Why that's important for HIV is that the latent reservoirs, the latent virus that stays with infected people for a lifetime, is in resting CD4 positive T cells. So the only way to get rid of it is you need a way to target it in vivo, and we've been able to do this. And this is now moving into macaques to look at, can we chop out the HIV provirus with Cre recombinase-like enzymes as part of a cure strategy for HIV. We had very high levels of recombination, both in spleen and in lymph nodes, with a single treatment of a low dose of RNA-LNP. So it was also fairly efficient. So our conclusions from this were that we could add targeting ligands, antibodies, pieces of antibodies, receptor ligand, proteins, just about anything, to the surface of an LNP and specifically target those cells to tissues, cells, and organs in vivo. We can deliver just about any protein that you can encode as an RNA. This can include therapeutic proteins that are deficient or therapeutic proteins that have an activity. It can deliver gene editing, gene modifying enzymes. Your imaginations can go wild with everything that can be delivered. When I give this talk, I'm always asked, well, what about the brain? And there's probably a variety of reasons why I'm asked that, and I won't discuss them now, but we can talk about it later. But I always put this data in because we've been able to target the brain with LNPs. So I don't know if there are clinicians in this office that have done their residency, but my absolute nightmare from residency was getting a patient admitted, usually Friday evening, who just had a stroke. And they would come in with this CAT scan. And what this CAT scan shows is that there's a region of dead, dying brain here. This is a middle cerebral artery stroke. And the patient comes in, they have some hemiplegia, they're not feeling great, and our job was to take care of them. The problem is what happens to those patients over the subsequent days. And this is typically what happens. This small stroke has now spread. And what this is the dying and dead brain cells here release inflammatory mediators. And they induce swelling in the brain, which compresses more brain and kills a larger area of brain. This isn't compatible with a meaningful life, and most of these patients don't do well. The problem is that clinicians have tried every anti-inflammatory and many different therapeutics with little success. So I work with some neuroscientists that have interesting models. And one model that they've got is that this is a mouse, and they can inject an inflammatory agent into one hemisphere of the brain. In this case, we're injecting TNF-alpha, which is a pro-inflammatory cytokine. And then we're delivering an anti-inflammatory mRNA. And what we wanted to know is, could we alleviate that inflammatory activity with an LNP that was directed to the brain? Now, the way we directed it to the brain is we added a VCAM antibody. VCAM is an endothelial cell antigen that's upregulated in the brain in the setting of inflammation. So what we're looking at here is that in red, this is a sham injected animal. They got the needle with no TNF. And you can see nothing happens. We're measuring vascular leakage in the brain. When we injected TNF, the inflammatory agent, and measured vascular leakage, it greatly increased on the side of injection, but not the opposite side. So we've got a built-in control. Here in red, we've delivered an LNP that's targeted to the brain with VCAM and encodes thrombomodulin, which is an anti-clotty, anti-inflammatory molecule. And as you can see, we've returned vascular leakage back to normal. We've gotten rid of all of the vascular leakage by this highly inflammatory agent. This therapeutic is moving forward into clinical trials as a new way of treating acute strokes to reduce the spread of the stroke and the spread of the damage. So I mentioned CAR T cells before. I'll give you a quick review of why CAR T cells are special. So this is a cytotoxic T cell trying to kill a tumor cell. It has a T cell receptor that recognizes a tumor-associated or tumor-specific peptide, and it can kill that tumor cell. The problem is that everybody has different peptides that their tumors make and that can be recognized. So the idea originally developed by St. Jude's was make a variant of the T cell receptor that has an antibody or another binding entity on the surface attached to the T cell receptor signaling. In this case, and these are the first two FDA-approved drugs, the antibody recognizes CD19. That's a marker on leukemia and lymphomas derived from B cells. This is FDA approved. It has efficacy rates in the 70% or so range, which for leukemias and lymphomas that were routinely deadly is an incredible achievement. The problem is that to make a CAR T cell, you have to do this. You take a patient who's got extensive cancer, you leukapheresis them. That's a machine that takes blood out, spins it in a centrifuge, withdraws the white blood cells, and gives the blood back. And it keeps doing that over and over for about 2 hours, and it takes out a couple billion white cells. You take those white cells and you infect them with a lentivirus that encodes the CAR gene. You then expand that T cell prep for typically 10 days to make trillions of cells. And then you take those cells and you inject them back into the patient. It has 70% efficacy. It costs about $500,000 per treatment because this 10 days of fancy treatment is done in a GMP cell culture facility that's only present in limited numbers of sites, mostly in the USA and Europe. Our idea was, if we could target lipid nanoparticles to T cells in vivo, and I showed you previously we could get expression of the mRNA, but in this case, we would target the LNPs and the RNA would encode a CAR molecule. In this case, we did a CAR molecule against a fibroblast-associated protein. And we used a model of cardiac fibrosis to determine. So what we saw is that with this targeting, we could get protein expression in 80% of the CD4s and CD8s with a single dose, so very efficient delivery. These are echocardiogram measurements. And what they basically do is they measure heart motion. So in gray, these are normal mice. In yellow, these are fibrotic mice that their heart function isn't working well. Sorry. So what you can see is that when the mice are fibrotic, the volume, the size of the heart increases. And in the bottom, this is the ejection fraction. That's how much blood is pumped out with each beat. You reduce the amount of blood. The heart gets stiff, gets bigger, it can't beat well. In red, that's a single treatment of T-cell targeted LNPs expressing a CAR directed against fibroblasts. And as you can see, the heart returned to normal. It's functioning completely normal with a single treatment. We stained the tissue for fibrosis, which shows up in red. The saline is untreated mice. The angiotensin phenylephrine are fibrotic mice. A single treatment with RNA-LNPs just about returned them to normal. We've also looked at targeting a variety of other cells and tissues. As an immunologist, I've always been interested in targeting bone marrow stem cells. There are thousands of genetic diseases in bone marrow stem cells. Nowadays, to treat them, the gene therapies do typically autologous bone marrow transplants, or they do gene therapy on their own stem cells. Both of those have fairly high levels of toxicity and are incredibly expensive, $2 to $4 million a treatment. We wanted to see, could we deliver lipid nanoparticles to stem cells in the bone marrow in vivo with an IV injection? And as you can see here, with fairly low doses, we were targeting 90+% of the bone marrow stem cells. Highly efficient. The way you test this is you treat the animal. In this case, it was a Cre-Lox animal. So we gene edited the bone marrow stem cells, and we followed them over time. And we can see we're out to 16 weeks in this, and 100% of the blood cells, so these are red cells, white cells, and granulocytes, are green. So they were gene edited with a single treatment at very high levels, approaching 100%. Now, the ultimate test is you do a transplant. You take the bone marrow from these treated mice after about 6 months, and you transplant them into autologous mice that have been irradiated or given chemotherapy to deplete their bone marrows. So we're replacing their bone marrow with the transplanted bone marrow. And as you can see here, excuse the complicated slide, but red is all 100%. And red are the transplanted blood cells, myeloid, lymphoid, a variety of different cell types. It doesn't matter. For all of them, 100% of the transplanted bone marrow cells were gene edited with a low dose of bone marrow targeted LNPs. Why is this important? We're developing therapies to treat sickle cell anemia with these LNPs. Now, sickle cell anemia is a fairly common genetic deficiency. The problem is it's started in sub-Saharan Africa. It's a way that the body developed to defeat malaria. The problem is that it leads to a very painful disease and a much shortened lifespan. 300,000 people a year are born with sickle cell anemia, mainly in sub-Saharan Africa, India, but across the world. You can't do ex vivo bone marrow gene therapy at $2 million a shot. That's unaffordable for the world. Our idea is that we've now got LNPs that will target bone marrow stem cells in vivo with 100% efficiency. Can we deliver gene editing machinery to fix that gene defect in sickle cell? So we've done this here, and this is a mouse model of sickle cell anemia, and we've delivered an adenosine base editor. So this is a Cas9 that's been turned into a base editor instead of a DNA cutter. And it changes a single nucleoside in the hemoglobin gene as directed by the guide RNA. It doesn't fix the hemoglobin, but it puts in what's known as a Macassar mutation that gets rid of the pathogenic activity of the sickle cell mutation. So you end up with a mutated hemoglobin, but it's a normal functioning hemoglobin where you don't get sickle cells. So you can see on the right, the arrows are pointing at sickle cells that are commonly seen in these animals. And on the left, after treatment, you no longer see any sickle cells. Here we're quantitating the gene editing ability of this enzymes delivered with lymph node bone marrow stem cell targeted LNPs. We're approaching about 90% of the bone marrow stem cells have been gene edited and the sickle cell gene knocked out. So this has potential to be an inexpensive and easy way of treating sickle cell anemia in the field. You simply have to walk up to somebody with hemoglobin S, give them an IV injection of RNA-LNPs, and potentially cure their sickle cell disease. And we're moving forward with help from the Gates Foundation to put this into clinical trials. So we can now perform in vivo gene therapy. And as I mentioned yesterday during my acceptance speech, a longtime passion of mine is equity of therapeutic developments and equity of drugs and vaccines. In vivo gene therapy will allow that equity. You don't need fancy hospitals and research centers to do gene therapy anymore. You can walk up to people, give them an IV injection, and cure their disease. This has applicability to thousands of other genetic disorders in bone marrow, in a variety of different cells that we can target. You can also use this to knock out stem cells as part of a bone marrow transplant for a variety of different cancers. The applications are just too numerous to mention. So I need to thank all of the people in my lab and all of the labs that I collaborate with to do these studies. Thank you.
Thank you very much to Professor Weissman. Thank you. Please take your seat. And finally, we would like to invite Professor Cullis to the stage to deliver his speech. Welcome, Professor Cullis.
Does the laser work? Okay, I think by now you should be convinced that lipid nanoparticles are actually quite important. So I'm going to give you a bit of a background on how it is that these lipid nanoparticles came to be. So you've seen some of the applications are becoming really quite stupendous from what Katie and Drew have described. So no, this thing doesn't work. Does anybody else have a laser?
So because the LED is too bright, so it's a little bit.
No, I see. It's not very bright.
No, this one. Next.
Yeah, I know, but I mean, it's very weak. Okay, the laser doesn't work. The technology has its limits. Anyway, you may have heard that this is again a little bit obscured here, but you may have heard that the vaccine revolution. This was back in about 2021. Some of the titles said the vaccine revolution is coming in tiny bubbles of fat. Now I have to say that I took this as a little bit of a simplification. This is both in the popular literature on the left, Bloomberg. And then on the right, even in the scientific literature, you are seeing these things referred to as tiny bubbles of fat. I wouldn't say I took this as a personal insult, but you know, there's so much more than tiny bubbles of fat. It really took 50 years for lipid nanoparticles to become an overnight success, as the term goes. So just going through my own history, you know, starting in about 1973, I started some basic studies on membrane lipids, not thinking really of any sort of application from a therapeutic point of view. However, in 1985, we started to try to develop lipid-based systems to deliver cancer drugs, to try and get them more accurately to where they're needed for individuals with cancer. And certainly is an ongoing problem that we still need to solve. Although we did do some work there, some success there. But starting about 1995, and so again in this theme of, these vaccines were not developed overnight. They were not developed in 10 months. They were developed in much longer periods of time. But starting in 1995, the development of lipid-based systems to deliver nucleic acid-based drugs to target cells in vivo. Now, the reason why some of these things, the format is wrong is because there's some translational difficulties. But anyway, in the beginning, I got my PhD in physics in 1972, and I decided that I had to do something different. The problem with physics is that the basic theory is for most phenomena is pretty well understood. And so you could do an experiment. I was an experimentalist. You could do an experiment, but if you got an answer that didn't agree with theory, then your experiment was probably wrong. And I found that rather depressing. It seemed that there's so many problems that were more interesting outside the field of physics. And I would encourage the younger members of the audience so I took a big transition here. I got interested in making these transitions is, I think, in the end, a good thing. I got interested, I knew a bit about nuclear magnetic resonance or MRI or other applications of that. I was awarded a fellowship to go to the biochemistry department in Oxford, and I knew nothing about biochemistry or lipids. It was rather a disorienting experience to walk into a biochemistry lab. Particularly the animal room was right next to the entrance. I remember going in, I thought, wow, this is certainly different. But my physics background turned out to be a good thing. This is me in 1975. You'll see some changes as the years have gone on. But what do you learn as a physicist? And you learn a technique for sure. The other thing you learn is that if you need an instrument that isn't available, then you build it. This is one of the precepts of physics. So when I got there, I found, well, while they'd been advertising that they had an NMR machine, really all they had was a magnet, and they didn't have the spectrometer to go with it. And so me and a graduate student in the lab, we spent a year putting this NMR machine together. So this was quite an effort. But we got it going, and then we tuned the NMR machine to phosphorus and started to look at the properties of lipids in biological membranes. Now, phospholipids have a phosphorus atom in the head group, and so that's the reason that we could detect lipids using phosphorus NMR. Now, to orient you here, a biological membrane is shown on the left-hand side. It's a tremendously complicated entity, probably the most complicated entity in nature, in biological systems anyway. And it consists of, a basic element of this is what's termed lipid bilayer, which is what you see in the middle. And so this is lipid bilayer separates the inside of a cell from the outside. And so without this, of course, a cell couldn't form, couldn't function. And so the lipid bilayer consists of lipids, as you see on the right-hand side, with a head group that contains the phosphorus, and then these acyl chains, the chains that repel water, so they're hydrophobic. And so there's about 100 or more different species of lipids in a given biological membrane, and but they have really different properties. If you extract the lipids from these biological membranes, you're probably wondering why this has anything to do with vaccines or anything else. I can tell you it was absolutely vital that we did these very basic studies. It's a great example of how basic science can inform can inform, give you techniques and approaches that really enable some advances. So some of these lipids, how do I go back here? Yeah, there we go. Will adopt, if you put them in water, when you extract them from a membrane, put them in water, they will adopt these non-bilayer structures. So what are they doing there? That was the question we were asking then. And then also the lipid composition on one side of a biological, so on the right-hand side here, on one side of a biological membrane is different from the other side. And so we said, okay, well, what consequences does that have? And can we drive lipid asymmetry? And what is the result of that? And so what we discovered was this phosphorus NMR, using our NMR machine that we'd managed to put together, we could detect which structures these lipids were in by the type of NMR signal that we saw. So we take the phospholipid that we extracted, we'd add water, they spontaneously adopt the structures. You can see there the bilayer structure, and we see this characteristic shape with a low field shoulder, high field peak by phosphorus NMR, whereas for the hexagonal phase, we'd see a peak with reversed asymmetry, and that was about a factor of 2 narrower. And these corresponded to bilayer and hexagonal phases, and we just interpreted this in terms of what you see on the right-hand side, which was the shape of the molecule. Lipids that were in the bilayer phase, for example, the area subtended in the head group region was the same as the area subtended by the acyl chains, whereas in the case of the hexagonal phase, the area in the head group was somewhat less. So this was a use of, from the point of lipid polymorphism, this was we published many papers in this area, but I'm not going to talk about those, although I could for about an hour or so, but probably not too conducive to passing on good information about vaccines. Anyway, we also used a different form of model membrane, as we term it, to look at the consequences of lipid asymmetry. And so we could make these liposomes or unilamellar vesicles, so in order to look at lipid asymmetry, we could add a little bit of what we termed an ionizable cationic lipid to that. Now, this ionizable cationic lipid turned out to be very useful, as we'll find out. But this was just originally we developed this lipid because at low pH, this is on the left-hand side, at low pH, now the pH 4, for example, this lipid became protonated, and so this is not very acid. pH 4 is about the acidity of a lemon, and then so it became protonated and positively charged, whereas at neutral pH it's net neutral. And so if we had a little vesicle here with a pH 4 on the inside and pH 7 on the outside, pH 7 being physiological pH, the neutral form could go across the membrane very easily, but it would get to the inside, become protonated, and it would stay there. And so we could generate lipid asymmetry. And so that was and we could do all kinds of things, make different shapes. If you have different lipid distributions, you can make all kinds of different shapes and all kinds of interesting consequences. But this is where we got a bit distracted. We got distracted by the drug delivery potential, because what we found was that we could accumulate drugs into these lipid nanoparticles, and I should say these are pretty small. They're about 100 nanometers or less in diameter, 1/100th the diameter of a cell. So they're really quite tiny. But we found we could accumulate a lot of drugs into these vesicles. They will go across in their neutral form, become protonated, and then positively charged or charged molecules don't permeate through lipid bilayers very well, so they'd be trapped on the inside. And if you do the maths here, at equilibrium, be 1,000, say for a 3 unit pH gradient, we'd have 1,000 times higher concentration of the drug on the inside versus the outside. So this is just doing this just to give you an illustration. This is a cryo-TEM, and just showing that for these vesicles, you can see the track around the outside of these vesicles on the right-hand side. That's the lipid bilayer. And then the crystal in the middle is doxorubicin, which is a common anti-cancer drug, which we could trap, say, 100,000 molecules of this cancer drug inside. So they made really ideal delivery systems to deliver these molecules more specifically to where we might need them. So we founded a company, again, another picture of me and 4 postdocs in my lab. This goes back to about 1992. We said, okay, well, this is really interesting. Maybe we can form a company to deliver cancer drugs more specifically to where they're needed. But actually, within a short time, within about 3 years, mainly because we had to raise money to keep this company going, and it was easier to raise money doing gene therapies than putting older cancer drugs in liposomes. So we started to work on delivering nucleic acid-based drugs. And so here we had to package them not in a lipid nanoparticle, but the problem was much bigger. For cancer drugs, all you had to do, even though this is very difficult to do, but is to get the drug to the site, to a cancer site, and then the drug will get into cells of its own accord. In the case of nucleic acid-based drugs, it not only has to get there, but you have to deliver it inside the cell, into the cytoplasm in order for the molecule to be able to do any work whatsoever. So we had a problem right off the bat. The nucleic acids are, as you're aware, negatively charged, very negatively charged, and so in order to encapsulate these nucleic acids in a lipid nanoparticle, we had to use positively charged lipids, which are termed cationic lipids. Now, there's no positively charged lipids in nature. They're really highly toxic. There's only net neutral lipids or negatively charged lipids. And so this was a, so in order to get past this problem, we went back to these ionizable lipids that we developed for our lipid asymmetry work. And we said, okay, well, maybe if we go down in pH to pH 4, we can encapsulate these nucleic acid-based drugs. This was antisense molecules at the time. Maybe when we come back up to neutral pH, we can maintain these molecules within the lipid nanoparticle, and that's turned out to be the case. And so we tried DODAP, which is the lipid that I indicated for which we were generating lipid asymmetry. Picture of Sean Semple on the left-hand side, who actually did some of these first experiments. And this actually worked pretty well. What we found was we could dissolve these lipids in ethanol, an organic solvent, together with what we might term helper lipids, phosphatidylcholine and cholesterol, and then mix that rapidly with a solution, aqueous solution at pH 4, containing the nucleic acid that we wanted to encapsulate. So conceptually, you can think about this. Here we're mixing these two streams very rapidly. The first thing to fall out of solution is going to be the negatively charged nucleic acid surrounded by the positively charged lipid. Now, if you do this fast enough, then these other lipids, the polyethylene glycol lipid, which is the one with the squiggly line here, will fall out of solution and encapsulate that and really trap it in what we term a limit-sized vesicle. So these are the smallest vesicles, or the smallest particles that are compatible with the molecular makeup of the entity. And so this was actually a bit of an advance, because these systems turned out to be relatively non-toxic because they weren't charged, or relatively low charge at neutral pH. They have what we term a hydrophobic core as opposed to an aqueous core, and they're really ideally suited to encapsulation of negatively charged macromolecules such as siRNA and mRNA. So we got excellent encapsulation efficiency. We could dial the size by changing the ratio of surface lipid to core lipid as a scalable and reproducible process. So I was at a conference in London in 2004, and I was accosted by a guy named Victor Katalin, who was the vice president research for a company in Boston called Alnylam. So Alnylam had been founded in 2002 to deliver something called small interfering RNA to silence a particular gene or silence genes in target cells, and they wanted to silence the gene in the liver. He said, we have a delivery problem. How can we get our small interfering RNA into the liver, into hepatocytes in vivo? And this was, I should say, I was pursued by Victor. He led me through half of Soho before we managed to find a place to discuss this. But it was really a very, very productive interaction with a 7-year collaboration that we went through with Alnylam to develop these lipid nanoparticles containing small interfering RNA to silence the gene in the liver. We had teams in Vancouver and teams in, as you can see here, teams also in Boston. And we started with this question. We had this very simple system that you'll see, as I've indicated, came out of some pretty basic research. The simple system that contained small interfering RNA, together with these ionizable lipids, this DODAP, surrounded by these PEG lipids in this kind of structure. And so could that do anything when we administered this to, when we were trying to deliver siRNA to the liver? So these particles get into cells by a process known as endocytosis. And so this is entailed a rather interesting design problem because we wanted them to get taken up by endocytosis, but then to get out of the endosome, and in a sense to destabilize the endosome. And so this really meant we had to design them so that they fused with the membrane surrounding the endosome at some vulnerable points in the endocytotic process. So they deliver the nucleic acid, the siRNA, into the cytoplasm. And so how could that happen? How could we destabilize the membrane, the lysosomal membrane? Well, this got us back to lipid polymorphism, our very basic studies on the structural properties of lipids, because it turned out that if you add an anionic lipid is a negatively charged lipid, such as the lipids you find in nature. If you add a positively charged lipid, you flip them over from a bilayer structure to these non-bilayer structures. And so as you can see on the right-hand side here. And so this gave us what we term a rational design process so that we could start to say, okay, how can we design these ionizable lipids so that it'll give us maximum disruption of the endosome and therefore maximum delivery into the cytoplasm. And so this is the kind of process that we envisage happening. On the left-hand side, we have our lipid nanoparticle containing our oligonucleotide or nucleic acid-based drug. The green is the ionizable lipid, and the low pH of the endosome, the pH of the endosome is reduced as it matures. Then these are converted to a positively charged form, indicated in red here, which induces fusion with the negatively charged lipids on the lysosomal on the endosome, which in the end results in delivery of the nucleic acid to the interior of the cell. Now we don't have any definite proof that this is what's happening, but it certainly correlates extremely well with the results. Now we get into more of the animal side of this. So we assess the potency of these systems using what was termed a factor 7 model. And so factor 7 is a clotting protein that's made in the liver, and it's also, and then it circulates in the circulation. And so it provided quite a convenient assay. We had factor 7 that would we had siRNA that would silence factor 7. If we injected that intravenously into a mouse, then we could assay for factor 7 in the circulation some 24 hours later. And so obviously if the siRNA that silences factor 7 was getting into hepatocytes, then we'd see lower levels of the of factor 7 in the circulation at the time of the assay. And so what we found was that not only was the ionizable lipid useful in terms of encapsulation and getting a low toxicity system, but the structure of the ionizable lipid could markedly affect the activity. And so this goes, all these ionizable lipids on the right-hand side here must look very similar, and they are. They've all got a tertiary amine and dilinoyl acyl chains, but they differ in one particular way, and that is the pH at which they become protonated, or the so-called pKa. Marcus Cipollini is a chemist I've been working with for the last 17 years now, is really quite a genius when it comes to making these systems. And so what we found was that there was this is a plot of the potency, it's 1 over the effective dose to get 50% gene silencing in the liver against the pKa of these lipids. And we found that a pKa of 6.4, if you're a 0.5 of a pH unit away from that in terms of the pKa, then the activity would go down by a factor of 100 or so. And so this is over the course of the collaboration with Alnylam, we went from a dose of about 10 milligrams or more of siRNA in order to get 50% silencing of the factor 7 down to 5 micrograms of siRNA. So really quite a dramatic change in potency, and this was without increasing toxicity at all. And so we had what's termed a therapeutic index of over 1,000. In other words, we could give 1,000 times higher dose before we'd see any toxic effects. And so this was ready to go into the clinic at this point. So the results of this approach in 2012 was that we could silence factor 7 in the liver. Now, these gene therapy approaches are remarkable in the sense that once we knew that we could silence factor 7, we knew we could silence any gene we wanted. All we had to do was change the sequence on the siRNA, and we would be able to do that. And so the clinicians at Alnylam said, okay, well, what we should do is develop a lipid nanoparticle containing siRNA to silence a gene called transthyretin. And the reason for this is that there's a disorder known as transthyretin-induced amyloidosis that affects about 50,000 people worldwide, a very nasty disease, actually. So this was a natural hereditary target to test this approach on. So this is HATTR. So transthyretin is made in the liver. It's then secreted into the circulation. And it usually forms this tetramer, but if there's a mutation, there can be many mutations, then it will form fibrils that presumably will distribute all through your body. And has really, these fibrils deposit particularly in nervous tissue and in cardiac tissue and have very nasty effects. There's really no effective therapy. It's usually fatal within 5 years of diagnosis. And so this is what you can see in terms of the prognosis. A pretty nasty disorder when you think of an individual who starts to have trouble walking when they're in their 30s, and they'll know what they're in for because they'll have seen a relative go through this condition. So a really nasty disease, but as I say, with the gene therapy approaches, it's very straightforward to develop a really targeted therapy. This has shut down the production of transthyretin. We should reduce these levels of fibrils in the circulation, and perhaps if we're really lucky, we should get some stabilization as the plaques, already established plaques, are cleared. So really a potentially simple solution to what's obviously a nasty disease. So this was moved into clinical trials in 2013. And so this is the results of a phase 1 study in healthy volunteers. Just pointing out that at about a dose of 0.15 milligrams per kilogram in these healthy volunteers, then the levels of transthyretin in the blood were knocked down by about 80 to 90%, and which lasted for about 2 or 3 weeks. And so on the basis of this, clinicians chose a dose of 0.3 mg of sRNA per kilogram body weight, given every 3 weeks intravenously for subsequent trials. So this is the results of a phase 3 study that was done, that ended in 2017. So on 225 patients, 148 of them getting the drug, the lipid nanoparticle containing the small interfering RNA that to silence transthyretin, and then 77 getting sterile saline as the control. And then the primary endpoint was a change in what's called the neural impairment score. In other words, a measure of the neuropathy that was induced by the transthyretin. And there's also secondary endpoints, self-reported quality of life, weakness, ability to walk, body mass, as you saw, it's a wasting disorder, etc. Now, this is where it gets quite spectacular, because this is looking at the results over the 18 months of this trial. And as you can see, the neural impairment score got steadily worse for those patients that were getting the sterile saline, whereas, if anything, the individuals that were receiving lipid nanoparticle containing transthyretin were getting better. So we're reversing the effects of a hereditary disorder. And so this was this result. This is the overall results for this clinical trial. An absolutely spectacular set of results, p-values that left absolutely no doubt that the drug was working. 10 to the minus 24, this 1 over Avogadro's number. So, you know, this drug really works. And similarly for other secondary endpoints, quality of life, again, p-values that were completely convincing. So it's a stabilizing and really quite a curative disorder, a curative therapy for what was previously a fatal disease. So I thought this is about as good as it gets for my career, being part of something where we have I've been part of a group that's had such an effect. But that was not. There were bigger things that followed. So this is the first FDA approval of a RNAi-based drug. It was a clinical validation of the lipid nanoparticle approach, and really dramatically demonstrates the power of these gene therapy approaches. But when Onpattro, as the drug is termed, went into the clinic in 2012, 2013, a company that I'd co-founded called Cullis, we said, okay, well, gee, we can't work on this drug anymore because it's in the clinic. You can't change things after it goes into the clinic. And so maybe we should see whether or not we could deliver messenger RNA, which is much larger than the small interfering RNA. And so we decided to see whether this approach that we developed to package the small interfering RNA using this mixing technique, dissolving the lipids in ethanol and then mixing with in this case, the messenger RNA in a low pH, a pH 4 buffer. And we got very similar structures coming out the other end. So this was, in other words, the encapsulation procedure really worked quite well. And we found that if we injected this into animals, into mice in this case, that we could then see, and this is using luciferase that Drew was referring to earlier as a marker enzyme, at 3 hours and at 6 hours, we could see that we were actually transfecting the liver using this approach. In other words, proteins were being made in the liver using these very similar, we did change some of the lipid compositions in order to get these results, but using very similar systems to what we developed for small interfering RNA. And so we've gone through a process here of developing more and more potent systems, part of that being improving the ionizable lipids, to get further and further improvements. Now, your liver makes, I don't know, 70% or so of the proteins in your body. And it doesn't seem to mind making a few more. And so if you inject, in this case, pigs, this is work done in, I guess, 2015, 2016, something in like that. But if you inject mRNA coding, in this case for erythropoietin, the liver will then make erythropoietin. This is in supraphysiological levels. Now, this is a pretty useful process when you think about it. Child is born lacking a certain enzyme, we can get the liver to make whatever. And so this was the direction we were going in. This is work done with Drew, injecting mRNA coding for a monoclonal antibody against HIV. And what was shown here was that these systems were very effective. They produced the, I don't have that slide in here, but they produce the monoclonal antibody against HIV and give protection to the animals against the subsequent infections. So you can actually get, these monoclonal antibodies being expressed in the circulation, and it has potential therapeutic benefit. But as I say, this is where we got quite lucky, because we were approached by Drew and to some extent Katie in 2012, in 2014, they had a problem that you've heard about, which was to okay, how do we get our messenger RNA coding for viral proteins into muscle and immune cells in vivo? And so obviously seeing the results that we'd been getting proteins expressed in the liver following an IV injection. And so the objective here, of course, was to take a viral protein, have messenger RNA coding for that viral protein, and then inject that intramuscularly to see whether if the proteins were expressed in immune cells, it would see a reasonable immune response. And you've already heard that was actually pretty, the results were fairly compelling. So this is just indicating we wanted to simulate a vigorous MHC1, MHC2 immune response. And as Drew has indicated, that was certainly observed. So the first example of this, and again, this is work done with Drew and his lab, the University of Pennsylvania, was to try and develop a Zika virus vaccine. And so here the messenger RNA in the lipid nanoparticle was coding for what's termed the Zika virus premembrane and envelope glycoprotein. So one of the proteins on the virus that you can see in the top right-hand side here. And this afforded total protection against the Zika virus. And so this is a test in an animal system. Indicate where we injected, or Party et al. Injected lipid nanoparticles containing messenger RNA coding for this premembrane protein, and injected that subcutaneous or intradermally, and then challenged at 2 weeks or 20 weeks with 200 PFUs of the Zika virus itself, and seeing total protection in these systems, as you can see here. The animals, the immunized animals, gave no evidence of infection whatsoever. So this really triggered a number of events. One of them was that we being acuitists, started to work with BioNTech in Germany. To develop a flu vaccine. This was about 2017 or 2018, 2019. And BioNTech in turn had been working, or was working, with Pfizer to develop a flu vaccine. And of course, when the pandemic hit, all attention was then diverted to making a vaccine for COVID-19. And so in this case, the messenger RNA that was packaged into the into the lipid nanoparticle, of course, was mRNA coding for the SARS-CoV-2 spike glycoprotein. And so the results of this trial, I mean, I'm sure everybody's aware, is now history. The results were announced in 2020, November of 2020, 95% effective. And I think we were all kind of stunned by that result. And that was consistent across gender, age, ethnicity, et cetera. And the vaccine was pretty well tolerated. At the bottom here, you can see it was anticipated they'd manufacture 1.3 billion doses by the end of 2021. I think they were closer to 3 billion by the end of 2021 and another 3 billion in 2022. It's been really quite an amazing process here to see this approved in various jurisdictions, and certainly played a major role in ending the COVID-19 pandemic. So I just wanted to say that this has been 27 years to develop lipid nanoparticles beginning, but really this is beginning with very, very basic science, trying to understand the roles of lipids and membranes, and that's really what led to these lipid nanoparticles that have had a pretty fundamental effect in terms of new therapeutics. So as Drew was emphasizing, though, the journey here is just beginning. These lipid nanoparticle systems are really, I term them the third generation of pharmaceuticals. The first generation being small molecule drugs, aspirin or cancer drugs, whatever. Second generation biologics, such as monoclonal antibodies. But the third generation is going to be these lipid nanoparticle messenger RNA gene therapies for protein replacement, vaccines, and gene editing therapeutics. So I looked up the number of clinical trials. I think that Katie had 150, I had 450, but there's a lot of clinical trials going on in this that are really pushing these approaches using mRNA and lipid nanoparticle delivery systems. We are going to see certain things coming along, again, as was indicated by Drew, one of them being a universal flu vaccine. This is just indicating if we take about 4 proteins associated with influenza and then have mRNA that's coding for all of them in a lipid nanoparticle that already affords protection against different strains going right back to 1934. So these are hopefully in the near future, we won't have to have a new flu vaccine every year. This is for atherosclerosis. It's got a little bit obliterated, the title here, but prevention of atherosclerosis. This is just gene editing on a protein in the liver that regulates the amount of low-density lipoprotein, or so-called bad cholesterol, in the blood, and you see you're getting a 60 to 70% reduction using this approach to gene editing to knock out that particular gene in the liver. So we can see like it's an immunization against what kills about 50% of us, which is atherosclerosis. So quite kind of monumental. This is a slide that's already been shown by Drew. But it began pointing out that, was this Drew or Katie? I'm not quite sure which one. I'm getting a bit mixed up here. But anyway, again, pointing out treatments for cancer. And there's so many new approaches now coming out of this for cancer. We can get the liver to make an antibody. We can do CAR T cell therapy, in vivo CAR T to try to eliminate the cancer. And then also the potential for cancer vaccines. So it's really a revolution in medicine that we're about to see. That's highly specific, highly personalized, and be developed very rapidly in times on the order of 2 or 3 months. So it's a big change. So this is the future. Really, as I said, quite remarkable in terms of the range of opportunity and being able to tackle in a fundamental way diseases that we really couldn't go after in an intelligent manner before. So what I've covered here is really the work of, I mean, literally hundreds, if not more, people. One of the things I've benefited from, as I tried to emphasize at the beginning, is really long-term association with my associates. We started off the company in 1992 with four postdocs or four people who have been postdocs in my laboratory. We're still working together today. This is people, Mick Hope, Tom Madden, et cetera, and others at 20 years or more, Steve Ansel, Ying Tam, Sharon Semple, Barb, et cetera. People at Alnylam, the Alnylam collaboration was enormously formative in this work. These things work in the brain, as Drew was indicating. Working with Drew has been revolutionary for us. And then my own lab in the biochemistry department at Oxford. So it's a biochemistry department at UBC. So it's this is it's been a real honor to be here and to receive the Tang Prize. I think what we all have to say here is that it's this in terms of the pharmaceutical applications that are being realized. In addition to the impact that the vaccine has made, this really is a big change in medicine, and one that we're all hastening to exploit as best we can. But it's really going to change the world in terms of the therapeutic potentials. So with that, thank you very much.
Thank you very much to Professor Cullis for your speech. Please take your seat. Thank you. And finally, we would like to invite back to the stage Professor Wang to give us the conclusion. Welcome.
Wow, it's really exciting to hear all three speakers talking about their, original goals and what they achieved and what the futures will look like. And since this is a 2022 Tang Prize laureates, and during our committee meetings, of course, we review many submissions. And finally, we chose these three laureates for the following reason. Here's our citation. It says, for the discovery of key vaccinology concepts and approaches leading to the successful development of mRNA-based COVID-19 vaccine. So at that time, we cited specifically on COVID-19. However, today's lecture gave us this future perspectives of many applications of this mRNA-LNP technology platforms. It can be used to counter infectious diseases, chronic diseases, cancers, and many others. So this is truly an exciting perspective for us. Now, of course, let me just say this contribution, I will phrase one of the nominators. It says, no other recent scientific discovery had a higher and more positive impact on humans than the COVID-19 mRNA vaccine. Now, of course, in the future, we can expect that many other applications will come forward. But today, the three laureates all have their stories, and I think I want to say a few words about them. For our young scientists, you see, all three of them have their unique stories. For Dr. Kariko, as you have heard, she has encountered many troubles over her career. Over the 40 years, she has been dismissed, demoted, and lost the jobs, and so on. However, she persist her interest and continue the research in this area. And then when I look at her CV, you know, before 2021, she obtained a few awards. But since 2021, each year, in 2021 and 2022, each year she had earned more than 30 awards a year. So it's a step function. Huh? So you can see that for young scientists, if you are in trouble or depressed, but don't give up. Just continue. Now for Dr. Weissman, of course, he is a successful model for us. But nonetheless, you have heard that when he started about this mRNA ideas and related things to some of his seniors, they smile and then politely says, oh, that's a good story, but don't do that, you know. So that's another example that if you want to be successful, stick to your ideas, to your imagination, but continue, okay? And finally, Pieter Cullis is a physicist. And he said, when he started, he knew nothing about biochemistry, just like me. Huh? And but physicists, they have a lot of talents. But what do you do to apply your talents to different things? Fortunately for us, he chose this fat, the lipid, I mean, to study and develop this wonderful technology of LNP, which makes the successful story of not only this COVID-19 mRNA vaccines, but I'm sure in the future for other stories as well. So for young scientists, I think you can learn from this. In Pieter's case, it's an interdisciplinary application of your mind. And but when you are down and out, don't give up. You know, those are the kind of lesson I learned myself. Okay. Now, of course, Tang Prize, especially in the biopharmaceutical area, because this is the area that only Nobel Prize has. Huh? So we are pretty successful in terms of the prize leading to the ultimate Nobel Prize. So I'm sure they are being considered in Stockholm right now. And I wish them luck. Perhaps in 2 months, we will hear that they will go to Stockholm for their next prize. Thank you very much.
Thank you very much to Professor Wang for the inspiring conclusion. And thank you once again to our laureates, Professor Kariko, Professor Weissman, and Professor Cullis. Ladies and gentlemen, this concludes the laureate lecture for biopharmaceutical science. Thank you for joining us this afternoon. Please don't forget your personal belongings as you leave, and we wish you a good day. 各位来宾，第5届唐奖得奖人演讲生技医药厂到这边圆满告一个段落。非常感谢您今天的参与，希望大家都有满满的收获。离场的时候，请记得您随身携带的物品，并且归还您所借取的同步口译接收器，取回您的证件。谢谢各位，也祝福各位平安顺心。